Good afternoon, everybody, and welcome to the Department of the Garrod Seminar Series. My name's Cyprian Broodbank, and for any of you whom I haven't met before, welcome. And I'm the head of department and will be chairing the remaining Garrod series for this term. And I wanted to start by saying, obviously, at this slight pivot point in terms of our subject matter, I wanted to thank Paul Lane and Laurie Bonner very warmly indeed for an extraordinary opening series of talks. Culminating last week in our seminar panel discussion on embedding Black history and archaeology in British schools, which was an absolutely extraordinary, inspiring and exciting event. So thank you to them. At this stage, the topic of the Garrod pivots slightly towards a series of talks on human origins, biological anthropology, and associated areas for the next 4 weeks. And 3 of those will be given by many of our most cherished and recent recruits to the ranks of the tutorships in this department. But next week's one, actually, just to advertise it so that you will know and have a reminder, will be given by Rob Foley, who's equally cherished, but perhaps not a recent recruit to the department, who will be giving the annual McDonald lecture at, 5 o'clock. Next Wednesday, in fact. We won't have a Garrod that week. So it's a full panoply of expertise from across the generations on all these exciting issues. And it's a great pleasure today to introduce and to welcome Dr. Amélie Beaudet. Amélie and I were joking that it was only about 3 months ago that she was giving a PowerPoint for the job of her life as one of the shortlisted candidates for our post in human origins. And we got it right, she got it right, she's here. We're incredibly delighted to have her with us. And of course, now she'll be talking about completely different stuff and telling us about what she really thinks. And she also asked very politely, if she goes over 40 minutes, will I stop her? To which she answered, she's got the job now. You've got the floor for as long as you need within reason. So Amélie, welcome, over to you. Why would welcome you here already? What do I mean? We're looking forward to this hugely. The title, it's always a bit silly for the person chairing and introducing to read the title, but ever more so now that they're sticking up there on the front of the screen in front of you all. So she'll be talking to us on Australopithecus from Sterkfontein, South Africa, and evolutionary species. You have the floor, and after that, please, if you could submit questions either by chat, or if that isn't getting through to me, by frantically waving, or any other form of communication in this Zoom age that you can manage. Laurie tells me she's here, lurking invisibly, and will also catch any questions. And she's also asked me to remind you, as I nearly forgot, that for any of you who can't attend this event live, there will be a recording that will be on the departmental YouTube channel, where you can also find recordings of last week's Garrod as well. So if you can't make it now, it will be up there for further consultation. Or if you like it that much, you can listen to it twice. There you go, Amélie, over to you.
Thank you very much for this introduction. Thank you for all of you to be virtually here for this talk. And I'm really happy to have the opportunity to speak about my research within the frame of the Garrod Research seminars. And so today I decided to speak to you about Australopithecus from Southern Africa, and more particularly from the site of Sterkfontein. And more specifically, I will be speaking about the variation within Australopithecus in Southern Africa and how we can interpret this variation. So what I'm going to do is to present a kind of a critical review of the traditional interpretation of this variation, but I will also speak about my own research during my postdocs in Southern Africa. And finally, I will discuss maybe new perspective or avenues of how to interpret this incredible variation and this very unique sample. So before going ahead, I just wanted to briefly introduce Australopithecus. And then, so it means also to speak about the big question in paleoanthropology, which is where do we come from? So what we know so far is that the potential earliest human remains is dated to 2.8 million years old and comes from Eastern Africa. But something interesting is that when we try to replace this remain into the hominin family tree that we can see here on the screen, we can clearly see that we still need to learn more, and there are still a lot of things that we don't know. And more particularly, and I think it's quite clear with this drawing, it's that we don't really know how the genus Homo that we can see here in blue is connected to any earlier hominins. So it means that we don't know exactly who is the direct ancestor of the genus Homo. However, so it is very likely that this direct ancestor comes from the genus Australopithecus. And that's why it's so interesting to study and look at the Australopithecus remains. So just very briefly, where do we find the Australopithecus remains and how old are they? So we can mostly find them in Eastern Africa with the two species, Australopithecus afarensis and anamensis. So Australopithecus afarensis is well known because it includes, for example, the specimen Lucy. But anamensis is also very interesting, and these two species are very well documented in the different sites in Eastern Africa. And in terms of chronology, so anamensis is older than afarensis. So there are more also remains, for example, from Chad with Australopithecus baryghazali, which is dated to about 3.6 million years old, and also two potential other species from Eastern Africa, but that are more less abundant than afarensis and anamensis. And these species are Australopithecus garri, which is dated to about 2.5 million years old, and also so Australopithecus derrimeda, dated to about 3.6, 3.3 million years old. But we can also find Australopithecus in Southern Africa. And so in that region, so these remains are attributed to two main species, Australopithecus africanus, dated about to 2.8 to 2.2 million years old, but also Australopithecus sediba, which is a bit younger and is only found in one site. However, there is also another hypothesis that then suggests that maybe there would be another species that would be Australopithecus prometheus, and this is what we're going to talk about today. So in this hypothesis, Prometheus will be a bit older than Africanus. So then the question is, how many Australopithecus species can we find in the fossil record? Because you can see here that the diversity is huge. And in the fossil record, this is very something that is intriguing. But of course, in that question and more generally, so in paleoanthropology, not all scientists agree on the fact that there would be up to 8 species within Australopithecus. So while we have a good agreement about the fact that, yes, there is Australopithecus afarensis and anamensis in Eastern Africa and Africanus in Southern Africa, for the other species, well, the debate is still there. So, for example, Prometheus could maybe be Africanus. And similarly for baryghazali and derrimeda, they might be potentially subsumed into afarensis. And even for Australopithecus garri and Australopithecus sediba, might be possible that they belong to another genus that could be either Paranthropus or Homo. So you can see here that there are still a lot of questions about the diversity within Australopithecus. And so this is what I'm going to discuss today, and I would focus more particularly on Southern Africa. So why to focus in South Africa? So for many reasons, but also what I think what is interesting here is that South Africa played a very important role. So in general in human evolution, but more particularly in the origins of Australopithecus. So in terms of historical context, this is in Southern Africa, well, in South Africa, where that the first Australopithecus specimen was found by Raymond Dart and published in 1925. And this discovery is very, very important because it is thanks to that fossil that then people started to admit and think that human origins were in Africa. And I think the context is also quite interesting because we are in 1925, and at the same time, so there was the Pilton man in the UK that was still considered as a valid kind of missing link. So the fact that Raymond Dart could find fossils that could be related to us in Africa was very, very important. And so this specimen is the tongue child, so it comes from the site of Tongue in South Africa. And so Raymond Dart then attributed it to the genus Australopithecus and the species Africanus. But since that time, so there were, of course, more and more discoveries, and more particularly in one area in South Africa that is known as the cradle of humankind. So in this area, there are different sites, paleontological sites, that are very close to each other. And some of them have provided a lot of fossil remains attributed to Australopithecus. So this is the case notably of Sterkfontein, but we have also the site of Malapa, where so Australopithecus sediva was found. And beyond the cradle of humankind, there is of course the site of Tongue, but also the site of Makapansgat, where a lot of specimens of Australopithecus were found. So here we'll speak more specifically about the site of Sterkfontein. So first of all, because this site have provided a lot of fossil remains. For example, so in one of the stratigraphic units, there were more than 800 hominins found, all attributed to Australopithecus. But also because the sequence here, well, the deposits document a very large period of time, and almost like the 3.5 last million years of human evolution in Southern Africa. So the site of Sterkfontein is looks like the picture that you can see on the screen. So there is the part of the cave that is exposed to the surface, and this part then is known as the Sterkfontein Formation. But there is, well, there are also localities that are underground, because the Sterkfontein caves, they are part of a karstic system. So it means that a system with different cavities related to, well, connected to each other. So underground, we can find other localities such as the Geographic cavern. So I will be speaking a bit about this later. And also the cavity where Littlefoot was found. So speak more about this. So this is the chronostratigraphy of the site of Sterkfontein, and more particularly of what we call the Sterkfontein Formation. So you can see that there are different units that are known as what called as members. So from the oldest to the youngest, we have the member 1, 2, 3, 4, and 5. So there are only, so member 2, 4, and 5 have yielded some fossil remains, well, for now. And so member 2 is the oldest. So this one has been dated to about 3.7 million years old. And this is where the specimen Littlefoot was found, which is the most complete Australopithecus skeleton known at the moment. And then we have member 4, which is dated to about 2.8 to 2.2 million years old. And this is where in this unit that we can find most of the fossil hominins, because in the member 2, so Littlefoot is the only hominin, surprisingly. And then we have the member 5, which is a bit younger, and there's no Australopithecus in that unit. So as I was saying earlier, there is another locality which is underground, and that is named Geographic cavern. So this locality is quite interesting because there are a lot of fossil remains there, all attributed to Australopithecus, but it seems that this locality is quite old. So actually the dating was about 4 million years old, so which means potentially older than Littlefoot. However, so more recently, scientists started to discuss and maybe debate this date, but at least, so even if it's not older than the member 2, it's potentially the same age, so still quite old. All right, so now we are going to do a little bit of history, but very briefly, just to explain to you then how the new findings could really impact our understanding of Australopithecus. So after the discovery of Tongue, then Robert Broom started to excavate at Sterkfontein. And so he could find the first adult Australopithecus specimen, which is the one that you can see on the screen, which is badly crushed, but still this discovery is quite important. And so at that time, he thought that this specimen was really different from the specimen of tongue. So he decided to create a new genus and a new species that would be Paleoanthropus transvalensis at Sterkfontein. And then a bit after, Raymond Dart started to work on the fossil specimen from Makapansrat, so not from Sterkfontein. And so here again, he thought that these hominins, they are a bit different from tongue and also different from Paleoanthropus transvalensis. So he suggested that then we should have a new species of Australopithecus that would be Australopithecus primitivus. But then finally in the 50s, John Robinson decided that all of these hominins found at Sterkfontein and Makapansrat at Tongue, they should be lumped into one unique Australopithecus species that would be Australopithecus africanus. So here it's quite nice to see, well, quite interesting because that reflects well the different theories in paleoanthropology that sometimes you are more like you want to split your sample in different species, or then at some point you just decide to lump all of them into one species. And so shortly after sometimes after the decision from Robinson to lump all the specimens into one species. So Lockwood and Tobias worked on the fossils from Sterkfontein and more specifically from the member 4. And because there are a lot of fossils there, they could start to think about the variation within Australopithecus. And then they realized that there is a very huge variation that they call polymorphism within that sample. It's quite surprising because it's like all of these hominins, they come from the same stratigraphic unit from the same site. And they even suggested that perhaps we have more than one species within that sample, but they refrain from naming this species. So they just say that perhaps we have more than one and that's it. But then quite at the same time, and then a bit before, Ron Clark started also to study this material. And he thought that there would be definitely more than one species at Sterkfontein, but also at Makapansrat. And so last year, Ron Clark and Cathy Kuhlman published a paper about Littlefoot. And then they suggested that this second species should be Australopithecus primitivus. So just briefly, what does it mean? So according to Ron Clark, we have in Southern Africa, more particularly at Sterkfontein, we have Australopithecus africanus, but also a second species that would be more like Paranthropus-like. So it means with some similarities with another group of hominins. And then he decided to name this second species as Australopithecus primitivus, so according to the previous paper from Raymond Dart. And interestingly, here it means that at Sterkfontein within the member 4, he suggests that there would be two species living at the same time and in the same environment. So they were kind of coexisting. So, but after all of these key papers and discoveries, there were more and more studies studying the variation at Sterkfontein, more particularly in member 4. And it's very interesting to notice that all of these studies, they converge toward the same conclusion that yes, there is variation. And what we can see is that a kind of a mosaic of human-like, but also Paranthropus-like, like Ron was saying, and also ape-like features within that sample. So the variation is real. It's not only like one scientist or two just thinking that, yes, that might be variable. It's just something that we can see and measure. So now the next question is, but how to interpret this variation? And I think this question is not only about Sterkfontein, it's also about, in general, in paleontology, how can we interpret variation? So of course we can say that there is more than one species, but there are other factors that we need to consider. So one that is very important is the sexual dimorphism, so which means that maybe it's because we have males and females within the same sample that it seems that there is variation, but in fact, it's just a normal variation within a species. The problem is that it's very difficult with the fossil sample to try to identify the specimen that would be males or females. So of course, we can use modern primates such as gorillas and chimpanzees to try to then see the different patterns or how the males and females differ, but we will never really know what is exactly in which extent they differ between them. And I think a nice example is the example of Mrs. Place. So this is the skull you can see, the cranium you can see here. And Mrs. Placer was found for member 4, and it's a very nicely preserved cranium. The thing is, there is still a debate to know if this specimen from Sterkfontein is more a male or a young male or a female. So even if with a very nicely preserved specimen, it's very hard to say if it's a male or female when we speak about hominins that are so old. The second concept or factor to consider is the geographical variation. So that sometimes when the specimen come from different areas, we might have to think about the effect of geography. But here it's not the case, of course, because all of the specimens, they come from the same site and the same, for most of them, from the same unit. So another possibility would be what we call ontogeny, which means the possibility that there are some juveniles in the sample. But here it's not, well, because we have a lot of materials, so this is something that is quite unlikely to bias our appreciation of variation. However, what is quite interesting here is that for Australopithecus africanus, the type specimen, so the specimen we use to define this species is a juvenile because it is the tongue child. So the problem is that when you use some criteria or morphological features on a juvenile specimen, then it could be possible that then when the specimen grows and become adults, maybe these characters can be modified. And then that's become very difficult to say that new specimens that are adults could be compared to the juvenile specimen, and then we can prove that they come from the same species. And then the last factors or potential source of variation could be the pathologies. So here again, cannot explain 100% of the variation because we have like a lot of specimens, and for the craniodontal specimens, so it's not the main factor. However, it's interesting to think about some of the post-cranial remains, so partial skeletons that were found at Sterkfontein that may be affected by some pathologies. And then potentially that could bias also our appreciation or description of the morphology, but also of the function and such as locomotion. Okay, so now this is the main background and history about the variation within Australopithecus at Sterkfontein. And so from that background, when I started my postdoc in Southern Africa, then I was thinking that maybe can kind of revise this unique sample by using new features and new technologies to try to learn more about the morphology of Australopithecus. So what I've been doing for the last years is to try to select some characters in the anatomy of Australopithecus that could tell us more about the variation, but also about potential different species or their biology. So I looked at different features from the cranium, so more particularly the brain, but also the brain case, and also the inner ear and the vertebra. So I'm going to show you the main results of these studies and how we can then interpret them within this framework of variation. So first of all, about the endocranial structures. So I was mainly interested in the brain because the brain is a kind of an interface between the hominins and their environment. So it can tell us a lot about their behaviors, but also we can also find some features that seems to be specific to some species. So that might help in understanding the diversity. And I also studied the brain case because it seems that more specifically in the cranial vault, the bone can show some features that are specific to some hominin species. For example, we know in Homo erectus that the cranial vault is thick and the organization is quite specific. It's different from Homo sapiens. So that's why I started to look at these two structures. So first of all, about the brain. So I was interested in looking at the brain organization. And I used the specimen from member 2, which is little foot, and also more specimen from member 4. And I tried also to select some specimens that were previously attributed by Ron Clark to Africanus, and also the second species that would be Prometheus. So it seems that, if we look at the brain organization, that this sample is quite homogeneous, that there is no huge variation. So here what I did is to look at the imprints of the brain on the inner surface of the brain case. And so from that imprints, I was able to identify the different separations, we'll say, or different areas in the brain. And so it seems that for the frontal lobe, more particularly, which is the anterior part of the brain, the organization is quite similar from one specimen to another. So we have an organization that is more like in extant chimpanzees, and it's different from extant humans, but this variation does not, this organization does not vary within the member 4 or even between member 2 and member 4. However, there is another part of the brain that is quite important. This is the visual cortex, which is at the back of the brain. And unfortunately, so this part of the brain is not really these imprints are not very well preserved. So I could not really check the hypothesis that maybe this part is also quite similar or not. And so that's something that maybe it would be possible to do with more specimen in the future. But speaking about the brain's organization, there is nothing that could tell us that there is a very important variation or unusual variation. And then so I started to look at the brain vascularization, and more particularly the imprints of the middle meningeal vessels on the inner surface of the brain case. So here again, the patterns, if we compare the Jakovic specimen, which is 4 million years old, and also member 2 and member 4 at Sterkfontein, so the pattern seems to be always the same, except that what we call the middle branch of this then pattern sometimes can connect to the anterior or posterior branch. So, but that is not unexpected because, for example, in extant humans, we know that we can observe this kind of variation. So here again, such as for the brain organization, we have something that is quite stable and the same pattern for all of the hominins. And then, so related to also this question of brain vascularization, I could look at the size of the carotid canal. So this is through that canal that the blood goes to the brain. And then I could compare the size of this then canal within the Australopithecus and also compare this variation to extant human and extant chimpanzees. And for these studies, was quite interesting because I had more specimens, especially from member 4. And here it was clear that this size variation was absolutely compatible with what we can observe in extant chimpanzees. So there is nothing that could maybe support the idea that there are more than one species. And then about the cranial vault, so as I was saying earlier, so there are some features that seems to be quite specific to some hominin species. So I looked at the thickness, so to look at which part of the cranium is thicker, and also at the organization, the microstructural organization, which means the proportion of the different bone tissues. So what I could see here by working on the specimen from Jakovic and also specimens from member 4 is that we always have the same pattern with the frontal bone being quite thick and also the parietal eminences being quite thick. And if we look at the bone tissues, we can always see that the spongious bone is quite thick and the compact bone is more thin. And so, and even speaking about the thickness, so we always have a variation that is compatible with what we could observe nowadays within extant human or extant chimpanzees. And then, as I was saying earlier, so I also looked at the inner ear and the vertebra. So why did I choose to look at these structures in particular? It's because they can tell us something about the locomotion, so the way the Australopithecus could move in the environment. And I choose to look at the inner ear because this is the organ of balance. So it's very important for locomotion, and also the vertebra. So if we still start with the inner ear, so here again, I could have quite a number of specimens that is quite important. So I could really look at the variation, especially within the member 4. And I could have like specimen from Jakovic, member 2, and member 4. So all of the different Australopithecus from Sterkfontein. And here the result is a bit different from what I could see from the cranium, which is quite interesting. So here it's a principal component analysis based on the morphology of the inner ear. And it seems that when we look at this morphology, more particularly within member 4, we can see 2 different morphogroups. So it's not 2 different species because you can see the variation is not that big. But it seems that some specimens, they can show more human-like features, whereas others can be more similar to the chimpanzees or Paranthropus. So there is variation that we can quantify here, notably in member 4, but also as compared to, by comparing the different Australopithecus at Sterkfontein. And the same thing seems to happen also with the vertebra. And so in this study, I focused on the first cervical vertebra, which is the atlas, because it's the interface between the head and the rest of the body. So for locomotion, this small bone is very important, in fact. So I could compare the morphology of the first cervical vertebra of Australopithecus that is quite well preserved, and as you can see here on the screen, and also one specimen from member 4, which is more less complete. But then I could compare the morphology, and here again I could run some statistical analysis, and what I could see is that these two specimens, they look a bit different. And more particularly, if we look at the articular facets, the morphology is definitely different. So it means that the way they were moving their head, or at least the head mobility, was different between the specimen from member 2 and the specimen from member 4, which are a bit younger, geologically younger. So the fact that we have different signals, depending on if we study the cranial structures or the structures that are related to locomotion, it's quite interesting, because it means that there's something that is quite intriguing, saying that maybe depending on the features you are looking at, you will have a different conclusion in the end. And that's why I think it's very important to combine the different structures to have different results. And quite interestingly, there was a paper recently submitted, so it's a preprint, so it's not published yet, but it's a paper from the Forney and colleagues, and they were studying the sacrum of Australopithecus within member 4. And it seems that based on their analysis, so here it's also statistical analysis based on the morphology, they could find that there is a variation, and they even suggested that based on this morphology, they could have 2 species within member 4. However, so some years before, the same team did exactly the same study, but on teeth, and more particularly the surface in between the enamel and the dentine, because we know that it's quite interesting, because we can use it to discriminate among the different species. And they found at that time that there was there was a variation, but that this variation cannot justify the coexistence of 2 species. So the same team then could realize that, could then provide some conclusions that are very different. So it means that there is definitely something going on between the cranial features and the post-cranial features. So now the question is, but how to interpret this? How to explain the fact that we have, depending on the structure we look at, we have different then conclusions. So here's this part is more about different ideas and theories and hypotheses, and I would be really happy to discuss all of them at the end of the presentation. So this discussion and this hypothesis, they are more related to what we say ecology and evolution. So first of all, about ecology. So if we think about it, the fact that there would be more than 1 species at Sterkfontein, more particularly in member 4, then it means that there are 2 species living together, living at the same time, at least, and in the same environment. And then so it would mean that they would have different ecological behaviors, because otherwise, how can 2 species then use the same resources in the same environment and still being 2 different species? So that's a very important question. It means that we need to explain how is it possible from an ecological point of view. So maybe the fact that we find some signals in the postcranial anatomy, or at least in features related to locomotion, could be explained by the fact that these hominins, they had different locomotor or ecological behaviors. But of course, we need more specimens to really try to test this hypothesis. And similarly, I think that's something we really need to do to try to figure out what is going on with Australopithecus. It's to consider that the hominins at that time, especially at Sterkfontein, they were not the only primate group living there. There was another group that we call papionins. So they include, for example, the baboons today. And the Australopithecus and well, the hominins and the papionins, they could be found in the same stratigraphic unit at Sterkfontein. So they are in stratigraphic association between hominins and the papionins. And then we can ask, but if they were living in the same environment and the same time, maybe we might be able to see if there are any similarities in the way these primates, they were reacting or overcoming some changes in the environment. So the different evolutionary pathways between the two different groups, we should be able to compare them and try to explain how they were evolving this way. And interestingly, here again, so with Australopithecus, we have the possibility that there is more than one species, but it's exactly the same with the papionins, so with the other primates. And because especially at Sterkfontein, we have this genus that is called Parapapio. And within Parapapio, and more particularly the Parapapio from number 4, there might be up to 3 species, which is even more than with the hominids. So we will have like Parapapio genesi, bromai, and whitey. So if it's the case, if it's true that we have so many species, then there should be maybe speciation event at some point at the history of Sterkfontein. And then, so the thing is, but can we find any evidence of this speciation event? However, just like for Australopithecus, for Parapapio, there is also a big debate about, but maybe we may have only 1 species. And maybe the fact that we find 3 species, it's because of the variation, but potentially this variation is compatible with the idea that there would be only 1 species. So this is something I could study during my PhD. And it seems quite clear that we have also variation within this group. And that if we need to understand what happened with the hominids, we need also to understand what happened to that group. So try to compare the different groups of primates. So the other hypothesis I wanted to explore here is the possibility that at Sterkfontein, and more particularly in the number 4, we have a temporally mixed sample. Because if we look at the deposits from number 4, so they are really huge. It's a very big stratigraphic unit. And this unit, well, the deposits, they span a time of about 0.6 million years old. So it's a very long time. So then it means that maybe time would be another factor to consider to explain the variation within Australopithecus. And so at some point I was thinking, but that would be interesting to compare this variation in South Africa with the variation of Australopithecus in Eastern Africa. And interestingly, there are a lot of specimens of Anamensis and Afarensis found in different sites, so like Kanapoy, Aliabe, Laetoli, and Hadar. And some authors, more particularly so Kimball and colleagues, they were thinking that maybe what we can observe is like what they call an energetically evolving lineage, which means that the variation we observe, it's because there is a very long period of time. And of course, there is no, like the hominins, they were evolving, like it's not like a stasis during, I don't know how many million of years. So because we only have, when we find fossils, we only have an idea of the hominins that were living at one place and during one specific time. So it's not reflecting really the reality because these hominins, they were evolving as well. So that's the suggestion, so that perhaps in this sample, we observe variation because it's just like evolution. And the question is well, maybe it's the same also in Southern Africa. The fact that at Sterkfontein, we have different period of times and we have this huge deposit in number 4, maybe it's because also we may observe an evolving lineage and we may observe the evolution of Australopithecus at Sterkfontein. And interestingly, I could find in one of the papers from Mogicetti and colleagues, this sentence that I think is interesting when we compare the Eastern Africa and Southern Africa, because they are saying that the Sterkfontein number 4 has generally similar levels of variability when compared to Afarensis, a species that shows the influence of temporal variations. So it means that, yes, there is one of the, perhaps the main factor that may explain this variation could be time. And so that's why the idea here I wanted to explore, it's really about the concept of evolutionary species. So it's just saying that maybe it's not about having one or two species. Maybe what we have here, it's an evolutionary species. So it means we have like a lineage with ancestors and descendants. And there is no clear like threshold or boundary between the two. So perhaps that would help to use this concept of species when we interpret the Southern African sample. And so by preparing this presentation, I was thinking that would be a great idea to finish this presentation with a quotation from Armélie Beaudet. And because I thought that this sentence would perfectly summarize all of these debates and questions. And so the sentence is saying that identifying species in human evolution is notoriously difficult and controversial. And to some extent, this arises from the desire to apply the biological species concept. So it means the fact that the formation of reproductive barriers between gene pools. So it means that trying to apply the conception of species that we have today with living specimens to the fossil records. And so as it concluded, so this is clearly impossible to observe directly. And I think that's why it's very interesting to speak about one, two, or maybe the one species or two species. But I think what is even more important is try to find a language, a common language between scientists that would help in communicating. So maybe it's not that important that there are two species, because maybe we cannot use these species like using the same definition as today. We need to use a slightly different species definition that would maybe help solving all of these debates. So I would like to sincerely thank all of my colleagues from Southern Africa, but also Europe and US and all over the world for all of this, well, for the past years when I tried to develop all of these projects and also the different funding I could get. And finally, I would like to thank all of you for listening to this presentation, and I would be really happy to answer your questions.
Thank you, Amélie. Thank you very, very much indeed. That was a fantastic presentation and many, many questions, I'm sure. I'm just having a quick look at the chat to see if any are coming in yet. Do you please send in your questions? Or otherwise, excellent, one from Rob. That's fantastic, Rob, fire away. Hello, Rob, did you get that?
Unmute. I always click the wrong button when I. So, Amélie, thank you very much. That was a lovely talk. I have to say, no matter how long I've worked in paleoanthropology, I've never really understood what's going on at Sterkfontein, which seems to epitomize all the difficulties of the subject in one site. And I thought you brought great clarity to that. I fully agree. I think in the end, we have to work with the evolutionary species concept. It's the only one. And I just sort of want to pick up something about what you're showing is mosaic evolution across this, in a sense that the result that the teeth show one thing, the locomotion show the other. And I'm just thinking about, if one brings in Australopithecus, which has a bizarre locomotion, you start to think across the range of things we find. The idea that actually what's happening in the Pliocene and amongst the Miocene and Pliocene is basically the way the apes are coping with the problems of changing environments is through locomotion, and not much else is changing. And you could even, if you wanted to be grand, go out and talk about Australopithecus or other things. So I just wonder if you have any comment about this, the idea that this is that what we see as a part of human evolution, this evolution of bipedalism, is really just a special case of a much broader phase of let's call it locomotory experimentation.
Yeah, I agree that it's and also the problem is that we don't have much of the post-cranial remains, especially for Australopithecus in Southern Africa and Eastern Africa. So it's always like well, for the same bone, we have like maybe three different analyses and three different conclusions, because we don't really know like the variation and how the entire skeleton looks like. And that's why I think also specimens like Littlefoot, they would really bring new having new insights into that kind of questions, because in this case we have like the upper limbs, lower limbs, and I'm not saying that it will clarify everything, but it's definitely something that will help to understand how the locomotion evolved.
Thank you. Can I encourage please send your questions in, because what usually happens is there's a huge pile up at the end and we can't take half of the best ones as they pile up.
I have a question.
Fantastic. Marta, fire away.
Yeah, Amélie, thank you so much. I love that talk. A question about the business about evolutionary species and anagenesis, because I actually think that there are two very, very different concepts. So one thing is to say that you have the transformation of one species into another through a non-branching process. That's simple. You know, the transformation happens in situ as a population evolves until it transforms itself into something so different that we would call it a different species. And another one is to think that there are evolutionary species in the concept by Simpson, but those should have independence and definite different destinies. That's part of the definition in Simpson. So conceptually, I mean, we have a problem in South Africa, right? Because there are trends, temporal trends that are convergent in the teeth of Africanus converging on Robustus. Then there are these distinctions that are expressed in some characters between Prometheus and Africanus and so on, and certainly Sediba, that takes us into a different space. So I wanted to know a little bit more of why you think that this is equivalent to what has been argued about anagenesis from Anamensis to Afarensis.
So I think it's, well, anagenesis cannot explain all the human evolution. So I think at some point there were anagenesis, and another point we had cladogenesis. And I think what we observe, the fact that we can't really identify a clear threshold between the Prometheus and Africanus, it's probably because we cannot really differentiate them, and because just it's an evolving species. So the fact that the characters are changing and they are not like clearly well, two different adaptations in two different species. And also I was also thinking about, so comparing the two because of also the temporal range. The fact that we have, like at Hada and the other sites, we have a long time, and it's quite also the same as Sterkfontein before. And the variation seems to be quite similar. I would say, yeah, we have the same kind of patterns. So that's why I was thinking that maybe we could compare the two. And I think the problem also at Sterkfontein, it's reason why the people are not really considering this, it's because of the chronology. Of course, we don't have like specific, member 4 especially, it's nightmare. And that's why I was initially thinking, so that what I presented, so paper I'm trying to work on. But really to finalize it, I would need a clear chronological context at member 4. But unfortunately, so it's a study that is ongoing. And for example, that would be great to know if some of the specimens that look a bit more Homo-like, if they are more recent than the others that are more ape-like. And because we have the information, we know exactly where they come from, but it's just like, it's a nightmare. So I think the stratigraphy now it's almost done, but now we need also datings, and then we'll be then able to see if this hypothesis is fine and we can falsify it or not. But I think this is the only way at the moment. Yeah.
Thank you. And they will need a whole afternoon to discuss these African ones.
Emma Pomeroy, I think a question from you.
Hi, yeah, thanks, Amélie. That was so interesting. I agree. We need to sit down and have an entire afternoon on this because it's so interesting. So I want to pose a bit of a devil's advocate question, which is slightly daft, but you mentioned the fact that the species concept is difficult. And I think actually if we think about the species concept, even in the modern world where we can observe species, it's problematic. And then if we think about the evolutionary species concept, it's problematic because where do you draw the line in something that is presumably, you know, there's not a clear cut between one and the other. So is another solution to kind of just talk in different terms And what might those different terms be if we sort of put aside the species concept?
I think that's a good question. And I think from my point of view, I would suggest to speak about chronospecies, perhaps. Just the fact saying that we have one population during one specific period of time and what place, and that's it. Because it's just, I think it's more, well, initially, the systematics and the way we are naming species was supposed to be easier for us as scientists to discuss, but now we realize that in paleontology, it's not absolutely not the case. Like we need to change that, and perhaps speaking about chronospecies would be great, but I don't know, it doesn't seem to be like a consensus among people. So in South Africa, there are people like Francis Thackeray who worked on the material for years and years. So he thinks that that would be the solution, that maybe it would be much easier to speak about evolution if we only refer to chronospecies and not about species, like using the specific species concept.
Great, thank you.
Other questions?
If there are no urgent questions, I want to pick Amélie on that one.
Go ahead, go for it.
Amélie, what difference would it make? At the end, we would group the fossils, give them a name, whether they are chrono, real, evolutionary, populations, etc. We'll need units of analysis, and it wouldn't make any difference, whatever we think that our units are. At the end, all the time, we're grouping them so that we can compare them, contrast them, see if they are trends, see if they're different in different places. And it doesn't really matter what each person's concept of the species is.
Yes, yeah, but we need, I think we need to use the same, in the end, the same term, well, same language to just, like, for example, if I speak to you about Australopithecus, then just speaking about Australopithecus, then it doesn't make sense. I need to specify, well, this sample from, I don't know, Sterkfontein, that is from the Pleistocene, because otherwise, how can we then describe the evolutionary patterns if we don't use units? But there won't be,
Exactly.
But it's much, for me, it's, then you don't, by speaking about chronospecies, you don't mean that there are different species like following the species concept that we are using now. So, I mean, it's more objective, I would say, that it's just a category. It's not, doesn't mean something. And I think also it would be, for example, like the paper saying, oh, I found a new species. I think that that's for me less interesting than the paper saying, I revised the material and then I found this trend in evolution. And if we speak about chronospecies and not if we don't put an emphasis on species concept, maybe that would be better. I don't know.
And when you find your big new Australopithecus in a Janovic cover, and you will publish and say, I've got a new chronospecies.
No, you just say, I found a new specimen from a site and from that time, and that's it.
I'm teasing you. We'll need 2 afternoons, 3.
There are many long afternoons ahead of us. I have, by the way, I hope you can't all hear grade 1 piano practice in the background when I unmute, because there's a lot of it going on. My apologies if you can. But I have a question from the floor. Amélie, would you be prepared to define your concept of chronospecies for an audience, not all of whom may be familiar with the idea, so that we know what exactly it is that you're, I'm sure it's bread and butter to half the audience, but not to everyone here.
Yes, a chronospecies, it's really saying that we have some specimens at specific time, like saying, I don't know, for example, I would say that the specimens from number 4, they form a chronospecies because they are found at specific time and a specific geographical context. So it's just to speak about a specific sample and not to, but it doesn't mean anything in terms of they can't interpret. So there's no, this meaning is, well, it's not that case.
That's really helpful, I'm sure, for many people. Thank you. If, while we're waiting for other questions to pile in, could I ask a really stupid question? It's a slightly heuristic one, and there's no real answer to it. But if your sample size were to increase 100-fold, what would happen? Would it multiply your problems or multiply your solutions?
something both, like it probably, because this typically what happened at Sterkfontein is that initially there were only few specimens, and then there were more and more, and maybe up to 800 hominins. And so initially it was quite easy, thinking there are 3 specimens that are from 3 different species. But then because we have more and more specimens, we have also a better idea of the variation. And so I think it will, at the same time, of course, it brings more questions, because then you start to think that, okay, this character is a bit different or varies or etc. But at the same time, it's also great because you can really speak about a character, but looking at a big sample. So I think we need, well, from my point of view, we need always more specimens, because otherwise we can't just say, okay, this species is like this, so I define this character, this character, this character, so without knowing that maybe these characters, they just vary within the species, or yeah.
Thank you.
Cyprian, I have a question. Can I?
Yes, I can't see you, but far away.
I'm there. Thanks, Amélie. Thanks for a great talk. It was really interesting, especially for somebody who doesn't know all about it already. So I'm, of course, thinking about the living primates and wondering about closely related species like chimpanzees and bonobos. Would they meaningfully be differentiated on your different measures? Because they are, probably have quite similar brain size, similar teeth, similar locomotion, yet are very different in their sociality and other things. Would they be differentiated?
I think that's it's supposed to be the first step before we look at the variation within the fossils. We need to look at the variation within the extant primates, and more particularly, so yeah, the chimpanzees and bonobos. And so, for example, in the brain, so I could see there is a bit of variation in the brain organization, but we won't see like a completely new structure happening like within the sample. So that will help to then interpret the fossil records. But like for the locomotions and all of these traits, so in fact, yes, we always try to look at the variation between these two living species to see how they vary. But typically for the locomotion, there is also another constraint, is that most of the time we also look at captive animals, and that would of course bias our interpretations. I think from that perspective, it's also not always easy to have a perfect sample representing like the wild chimpanzees and bonobos, then we can rely on to interpret the fossil records. But I mean, definitely that's something that's potentially the most important before interpreting the fossil record.
Enrico, I think comment.
Yeah. So I mean, I really enjoyed this, and it's really thought-provoking in a sort of in a broader way, because while you guys were discussing about the concept of species, wearing the hat of sort of culture evolution, I was starting to think about, culture evolution. There's this whole discussion about culture as species and how this is relevant to this, and it occurred to me, of course, that this sort of transition from species to chronospecies is similar to how archaeologists are no longer considering culture as, necessarily as ethno-linguistic groups, but as a marker for, yes, I'm referring to that culture as that place in that time, but not necessarily a specific group or entity, because it's a great way to summarize information, and it's a great way to have a sort of a tool so that you can refer to something rather than describing things in a certain way. But then I guess it follows that ultimately, depending on the type of question you might have, in the case of culture, you might actually really want to go and identify ethno-linguistic groups. And so similarly, I wonder, is the notion of chronospecies something that is a sort of a working hypothesis, or something that is a tool for now, but fundamentally, ultimately, you want to identify species, you want to look at this, and it's only because we are in this sort of transitional state where we don't have enough sample, we don't have enough information, that we're using this as a placeholder. But, or do you think that's entirely doomed, and it's by the nature of the data, by the nature of the question, it's always going to be a chronospecies? So my question really, if I phrase this as a question, is chronospecies is a fill-in for now, or is it something that you think it's going to be that's the way it is, and we should really not even try to argue at least for this kind of data set the notion of species. I don't know if I explain myself clearly.
Yeah,
know
I think
it's all over different places.
I think it's great. It's a really good question, and I think I really see the current species from a practical point of view. It's just that we need it, and just to test more evolutionary hypotheses, and that you don't really focus on the fact, okay, I have 1, 2 species, but more about the evolutionary processes. Well, maybe in the future we'll have more material, more new techniques, maybe DNA, or I don't know what, and maybe at some point we can revise this concept and think that maybe now we know how to define species in paleontology. So why not? But now, well, as we were saying earlier, that even with the extant, living mammals or primates, we are not always able to distinguish the species. So if we can't do it on living specimens, then on fossils, I don't know. I think we really need also this reference, and so that we have an idea of what is going on today with the living species, and then we can study the fossil materials. But yeah, why not? I'm sure that this concept would change, and we have more, I don't know, more hypotheses and new thought about it. Definitely, it's just suggesting, I don't know, maybe something, an idea to just at the moment, okay, we just use it, and then we'll see later how, yeah, maybe it will change.
And I think Rob would like to come in on that. Rob.
What? Sorry,
You've gone blank.
Gone blank. It's the problem of doing these things at home, because there's actually somebody heavily banging on the door. So I was just going to answer the door, but can I come back in 1 second?
Well, we have a brief pause there.
If there's another question, go to that and I'll come back.
Rob, I'll go.
I could fill you in on the fact there's now only 1.8% between Trump and Biden in Pennsylvania, and 8% of the vote still to go, which is about 700,000 votes. So that should, just from the longue durée to the immediate, and then briefly back again. I hope Rob isn't too delayed at the door. But Marty, you're at another door I can see, or you're there at the same door?
I'm in a different place.
Different door.
I'm back. Sorry about that. No, I mean, if I can sort of chip in, I think, I mean, I'm picking up from really Enrico's question about whether this is a sort of pragmatic solution. And I think, I mean, the history of the concept was very much a pragmatic solution, but it was also part of the, Ernst Mayer's and others people's idea, how do we just clean up the human evolutionary tree? And I think the problem is, I mean, I think that the species concept you've picked up on the evolutionary species concept, Amélie, is the perfect one, because it captures the idea of an evolving lineage. And I'm not sure you really need anything beyond that in order to do it. And if you do, I would be tempted to for something like the term lineage, which expresses the ancestral descent relationship. It doesn't exclude branching lineages as well. And it avoids this kind of slight confusion as to, introducing the species concept, which is a biological one, into something that is then an analytical one. So I think I would, I mean, I can see it as a pragmatic solution, but I don't see it as a long-term evolutionary concept, would be my view. I mean, you probably know, I think there are now something like 26 different species concepts formally defined out there. So you can pretty much choose one that matches your analysis.
Can I just chip in from the genetic point of view here?
Please, Lisa.
So I think whatever, we'll settle on or we don't settle on, I mean, it's going to be a pragmatic one, because if we're thinking about what the process of speciation really looks like, it's just too messy. Like it's not a lineage, it's not lumps of individuals, or it's not even a population. It is literally a network of gene trees, spreading and then connecting again and kind of meeting in the same individual and breaking up, and it's incredibly messy. I mean, we know from living species how, you know, any names that we come up is, you know, is essentially just convenience. So I'm very much with Amélie here. I think we need to, you know, just kind of find a term that communicates the limitations of you know, the concepts that we've come up with.
Amélie, would you like a chance to respond to either of the last two comments?
I think, and I agree also with Rob, it's that there are a lot of different species concepts, and so this one won't be, even if the evolutionary species is convenient concept at the moment, well, it doesn't mean that's the best and we should keep it forever. And I think also that, something that might be important here, it's the lineage. And so, yes, it's just, we need to find a way to communicate, and so that we all understand when you speak about some specimen and samples, and that maybe and speak about really about how evolution worked, and
Are there any final questions?
I'm just going to comment, if that's all right.
Peter, please, yes.
Just to say that the way that the species concept has been described here very much supports Enrico's point earlier about it being analogous to the cultural evolution view, because in that we have to define our units of analysis at the start of everything. It's almost the processes are understood commonly, but the units you're actually dealing with that to apply those processes to are sort of defined according to the question working with. And so it's actually slightly encouraging to hear that it would also be true in biological units of analysis.
I sense a lot of conversations going on with and outward from today, which is exactly as it should be. These presentations should be beginnings, not endings, and that's wonderful. So thank you very, very much, Amélie, indeed, for giving us a marvelous, incredibly stimulating talk. Lots of virtual applause breaking out all over. Before everyone leaves, could I just, again, particularly for anyone who arrived late, remind you that next Wednesday, not Thursday, at 5 o'clock, we have the McDonald annual lecture, which will be delivered by Rob Foley on the fourth handshake, something I don't quite have at the moment, but along an explanatory subtitle, and he assures us and explain what the fourth handshake is on the day. So be there and discover what that all means. And then the week after that, there'll be no Garrod the next day, but the week after that, Emma and Kat, correct me if I've got this wrong on the trot, 19th of November, Emma Pomeroy, correct? Yes. And on the 26th of November, Kat Coops. And after that, a very well-deserved end of term for everybody, because it's been quite something so far. So thank you all. Thank you, Amélie. And
thank you.
See you all soon. Well done. Thank you.
Thank you. Bye.